Assalamualaikum and a very good day. So in this video, we from Group 3 will present our presentation based on an article with the title Quantitative Proteomics of the CDK9 Interactome reveals a function of the HSP90CD37 PTEFB complex for BTEI-induced HIV-1 latency reactivation. So our group members consist of myself. My name is IQBAL HAKIMI bin ADZAHAR. Next, MUHAMAD NABIL NAAIM bin MALAKI and DANIAL ASYRAF bin SALIM. So moving on to the first slide, which is the introduction. The first point, which is the role of BRD4 and PTEFB in transcription. BRD4 stands for brodomain containing protein 4, which functions as the transcriptional co-activator and epigenetic reader as it binds to the acetylated histone H3 and H4. This binding of BRD4 promotes the recruitment of the positive elongation factor B, PTEFB, which are made of CDK9 and cyclin T1, which causes RNA polymerase II at serine 2 to be phosphorylated. This allows the transition from promoter proximal posing to productive transcriptional elongation. Next, the second point is the significance of HIV activation and oncogene expression. The mechanism for the HIV-1 reactivation and expression of various oncogenes such as CMYC, BCL, XL and PEX5 causes BRD4 to be one of the potential targets for therapeutic. The third point, which is the prodomain and extra-terminal inhibitors. Prodomain and extra-terminal inhibitors, along with the well-characterized compound JQ1, causes a competitive binding to BRD4's prodomains, which interrupts the interactions between acetylated chromatin and induce the formation of the TET superelongation, which lead to the reactivation of latent HIV-1. So the last point, which is the importance of CDK9 and objectives of the study. Based on the primary role of CDK9 in both HIV-1 and cellular gene transcriptional elongation, the study conducted by Wang in 2023 utilized SILAC-based quantitative proteomics to determine CDK9 interacting proteins under JQ1 treatment, attempting to discover the substitutes for the mechanisms of transcriptional regulation beyond BRD4 mediated recruitment.
Assalamualaikum warahmatullahi wabarakatuh. My name is Danial Asyraf and let's begin with the study overview and the method principle. This study aimed to investigate how CDK9 protein interactions change after treatment with the BET inhibitor JQ1. CDK9 is essential in HIV-1 transcription, making it a key target in HIV latency reversal strategies. By understanding its interaction network, the researchers hope to identify mechanisms that could help reactivate latency HIV. The method used was SILAC, or stable isotope labeling by amino acids in cell culture. In this approach, proteins are labeled with either light or heavy amino acids. JQ1 treated cells were labeled heavy while untreated cells were labeled light. After treatment, proteins were extracted, mixed and digested into peptides. They were analyzed using LC-MS, which separates and identifies peptides based on their mass to charge ratios, enabling quantitative comparison between conditions. Now, let's look at the application of LC-MS in this study. Using this method, the researchers identified 352 proteins that interact with CDK9. They quantified changes in how these proteins bind to the CDK9 after JQ1 treatment, revealing increase or decrease in interaction levels. This allowed them to map the CDK9 interactome and uncover how protein networks shift during HIV transcriptional reactivation. a key finding was the discovery of a new complex, which is HSP90 CDC37P-TFB, which plays a crucial role in reactivating latent HIV, making it a promising therapeutic target. Several tools supported this analysis. One of them is Proteome Discoverer 2.4, which is used for processing mass spectrometry data. The second one is UniProt for protein identification. The third one is Metascape, which is pathway for enrichment analysis, and the last one is Pride, which is to publicly share the dataset. Finally, let's talk about the societal and industry relevance of this study. This research supports HIV cure strategies, especially the shock and kill approach, which aims to reactivate and eliminate latent virus. It also advances our understanding of gene regulation, which is relevant not only to HIV, but also to diseases like cancer and autoimmune conditions. The discovery of a new druggable complex opens the door to developing targeted therapies. Importantly, the study also supports repurposing existing drugs such as BET inhibitors and HSP90 inhibitors, which are already used in cancer treatments for potential use in HIV therapy. Overall, this work shows how proteomics and bioinformatics can translate basic science into the real-world medical applications.
Okay, now I will explain about the advantage. First is the development of potential therapeutic strategies, whereas these are just a new way to find or develop drug to eliminate HIV-1. The discovery support the idea of using multiple target drug together for the viral interaction, especially since HSP90 inhibitor has already been tested in a clinical trial for lung disease. Next is the quantitative insight into the protein interaction, where this method provides specific measurable data on the change in the proteomic interaction. It clearly shows a striking increase in the binding of HSP19 and CDC37 to the CDK9 when cells are treated with the BET inhibitor JQ1. This evidence are also confirmed by the analyzing the detail of the mass spectra. Next is the comprehensive tool and systematic. It uses mass spectrometry based quantitative proteomic, especially silica, stable isotope labeling with the amino acid in cell culture, coupled with LC mass spectra. This allows for a very true and systematic way to study how protein complex dynamically associate and dissociate under different conditions, like when BET inhibitor are used. Next is the disadvantage. First is the complex data analysis. The larger data set requires specialized bioinformatics expertise and software for accurate interpretation, which can be time-consuming and challenging. Next is the detection of limitation, whereas the quantitative proteomic faces several detection challenge primarily due to sample complexity and protein abundance differences. Last is the resource intensive. It requires specialized mass spectrometry equipment and is costly reagent like the isotopic label and extensive sample preparation, limiting accessible for some lab. Okay, that's all from me. Thank you.
In this short video, we'll demonstrate how to use the UniProt database to retrieve protein information and explore 3D structures using CDK9 as an example. First, go to www.uniprot.org, a global protein database that provides detailed information and protein sequences, function and structures. In the search bar, type CDK9 human. Then click on the review entry with UniProt ID P50750. This will take you to the protein's main page, which contains all the relevant data. Next, scroll to the structure section. This is where you can view the 3D model of CDK9. Here, you can rotate, zoom and examine the protein's domains interactively. Then after this, you can go to the sequence and isoform. This is where you can click or search the FASTA link to view and download the full amino acid sequence for further use in analysis tool. Now you can click the download button, and as you can see, this is the view full format of the FASTA link for this CDK9 human gene. And that's it. In just a few steps, Uniprot lets you access detailed protein data and visualize structure all in one place.